Now we are going to learn about eye and retina. So first let's learn about fluid system of the eye. That is the intraocular fluid. Intraocular fluid. The eye is filled with intraocular fluid which maintains sufficient pressure in the eyeball to keep it distended. The fluid can be divided into two portions: aqueous humor, which lies in front of the lens, and vitreous humor, which is between the posterior surface of the lens and the retina. These are intraocular fluid. The aqueous humor is freely floating fluid, whereas the vitreous humor, sometimes called the vitreous body, is gelatinous mass. Gelatinous mass. Which one? Vitreous fluid, gelatinous mass. And held together by a fine fibrillar network composed primarily of greatly elongated proteoglycan molecules. Both water and dissolved substances can diffuse slowly in the vitreous humor, but there is little flow of fluid. Aqueous humor is continuously being formed and reabsorbed. The balance between formation and reabsorption of aqueous humor regulates the total volume and pressure of the intraocular fluid. Aqueous humor, iris fluid, flow of fluid, formation of aqueous humor, spaces of Fontana, canal of Schwann, ciliary body, diffusion of fluid and other constituents, filtration and diffusion at retinal vessels, then optic nerve. These are the portions of this fluid system. Formation of aqueous humor by the ciliary body. Aqueous humor is formed in the eye at an average rate of 1 to 2 ml each minute. Especially all of it is secreted by the ciliary process, which are linear folds projecting from the ciliary body into the space behind the iris where the lens, ligament and ciliary muscle attach to the eyeball. Cross section of the ciliary muscles is very important, and their relation to the fluid chamber of the retina eye is there. Because of their folded architecture, the total surface area of ciliary process is about 6 cm square in each eye, a large area considering the small size of the ciliary body. Surfaces of this process are covered by highly specialized epithelial cells, and immediately beneath them is a highly vascular area. So ciliary processes, formation of aqueous humor, vascular layer, ciliary muscle. Aqueous humor is formed almost entirely as an active secretion by the epithelium of the ciliary process. Aqueous humor is formed almost entirely as an active secretion in the epithelium of the ciliary process. Secretion begins with active transport of sodium ions into the spaces between the epithelial cells. The sodium ions put chloride and bicarbonate ions along with them to maintain electrical neutrality. All the ions together cause osmosis of water from the blood capillaries lying below into the same epithelial intracellular spaces, and the resulting solution washes from the spaces of the ciliary process into the anterior chamber of the eye. In addition, several nutrients are transported across the epithelium by active transport or facilitated diffusion. They include amino acids, ascorbic acid, and glucose. Outflow of aqueous humor from the eye. After aqueous humor is formed by the ciliary process, once again I am going to repeat that ciliary process. It first flows through the pupil into the anterior chamber of the eye. Pupil into the anterior chamber of the eye. From there, the fluid flows anterior to the lens and into the angle between the cornea and the iris. Then through the meshwork trabeculae, finally entering the canal of Schwann, SCHLEM, which empties into extraocular veins, showing that the spaces between the trabeculae extend all the way from the anterior chamber to the canal of Schlemm, SCHLEM. The canal of Schlemm is a thin-walled vein that extends circumferentially all the way around the eye. Its endothelial membrane is so porous that even large protein molecules as well as small particulate matter up to the size of red blood cells can pass from the anterior chamber into the canal of Schwann. Schlumm. Even though the canal of Schlumm is actually a venous blood vessel. So much aqueous humor normally flows into it, and it is filled only with aqueous humor rather than with blood. The small veins and that lead to the canal of the Schwann to the larger veins of the eye usually contain only aqueous humor, and it is called aqueous veins. Then intraocular pressure. The average normal intraocular pressure is about 15 mmHg with a range from 12 to 20 mmHg. Measuring intraocular pressure by tonometer, because it is impractical to pass a needle into a patient's eye to measure an intraocular pressure. This pressure is measured clinically by using a tonometer. The principle which is briefly explaining. The cornea of an eye is anesthetized with local anesthetic, and the foot plate of a tonometer is placed in the cornea. A small force is then applied to a central plug, causing the part of the cornea beneath the plugger to be displaced inward. The amount of displacement is recorded on the scale of the tonometer, and this is calibrated in terms of intraocular pressure. Regulation of intraocular pressure. Intraocular pressure remains constant in normal eye, usually within plus to minus mmHg of its normal level, which averages about 15 mmHg. The level of this pressure is determined mainly by the resistance to outflow of aqueous humor from the anterior chamber into the canal of Schwann. This outflow resistance results from the network of trabeculae through which the fluid must percolate on this way from the lateral angles of the anterior chamber to the wall of the canal of Schlumm. These trabeculae have minute openings of only 2 to 3 mm. The rate of fluid flow into the canal increases markedly as the pressure rises. At about 15 mmHg in the normal eye, the amount of fluid leaving the eye by the canal of Schlumm usually averages 2.5 ml per minute and equals the inflow of fluid from the ciliary body. The pressure normally remains at about this level of 15 mmHg. Then glaucoma causes high intraocular pressure, and it's a principal cause of blindness. Glaucoma is one of the most common cause of blindness. It is a disease of an eye which the intraocular pressure becomes pathologically high, sometimes raising acutely to 60 to 70 mmHg. Pressure above 25 to 30 mmHg can cause loss of vision when maintained for long periods. Extremely high pressure can cause blindness within days or even hours. As the pressure rises, the axons of the optic nerve are compressed, where they leave the eyeball at the optic disc. This compression is believed to block axonal flow of cytoplasm from the retinal neuronal cell bodies into the optic nerve fibers leading to the brain. The result is lack of appropriate nutrition of the fibers, which eventually cause death of the involved fibers. It is possible that compression of the retinal artery, which enters the eyeball at the optic disc, also adds to neuronal damage by reducing nutrition to the eye or retina. In most cases of glaucoma, the abnormally high blood pressure results from increased resistance to fluid outflow to the trabecular space into the canal of Schlumm at the iridocorneal junction. For instance, in acute eye inflammation, the white blood cells and tissue debris can block the trabecular spaces and cause an acute increase in intraocular pressure. In chronic conditions, especially in older persons, fibrous occlusion of the trabecular space appears to be the likely culprit. Then glaucoma can sometimes be treated by placing drops in the eye that contain a drug that diffuses into the eyeball and reduces the secretion of increasing the absorption of aqueous humor. When drug therapy fails, operative techniques to open the spaces of trabeculae or to make channels to allow fluid to flow directly from the fluid space of eyeball into the subcongenital space outside the eyeball can often effectively reduce the pressure. Entering into the next major topic, the retina. The retina is the light sensitive portion of the eye that contains cones, which are responsible for color vision, CC, and rods, which can detect dim light and are mainly responsible for black and white vision and vision in dark. When either rods or cones are excited, signals are transmitted first through successive layers of neurons in the retina and finally into optic nerve fibers and the cerebral cortex. Under low light conditions, vision is mainly served by the roots and this is called as photopic vision. You might have learned about this. Scotopic vision. Under well-lit conditions, the cones primarily serve vision and color can be easily be determined. This is called photopic vision. In this chapter, we explain the mechanics by which the roots and cones detect light and color and convert visual image into optic nerve signals. Anatomy and the function of the structural elements of the retina. Layers of the retina, which are arranged in layers from the outside to the inside as follows. Pigmented layer of rods and cones projecting to the pigment, outer nuclear layer containing the cell bodies of the rods and cones, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic layer, layer of optic nerve fibers and inner limiting membrane. Once again, pigmented layer of rods and cones projecting to the pigment, outer nuclear layer containing the cell bodies of the rods and cones, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic layer, layer of optic nerve fibers and inner limiting membrane. These are represented in a simple manner. So after light passes through the lens system of the eye and then through the vitreous humor, it enters the retina from the inside of the eye. After light passes through the lens system of the eye and then through the vitreous humor, it enters the retina from the inside of the eye. That is, passes first through the ganglion cells and then through the plexiform and nuclear layers before it finally reaches the layer of rods and cones located at the way on the outer edge of the retina. This distance is a thickness of several hundred micrometers. Usually, visual acuity is decreased by this passage through much non-homogeneous tissue. However, in the central foveal region of the retina, as discussed subsequently, the inside layer is pulled inside to decrease the loss of acuity. Then foveal region of the retina and its importance in acute vision. The fovea, FOVEA, is a minute area in the center of the retina, occupying the total area a little more than 1 mm square. It is especially capable of acute and detailed vision. Fovea. The central fovea, only 0.3 mm in diameter, is composed almost entirely of cones. Those cones have a special structure that aids their detection in detail in the visual image. That is, the foveal cones have especially long and slender bodies. In contradiction to the much fatter cones located more peripherally in the retina. Also in the foveal region, the blood vessels, ganglion cells, inner nuclear layer cells and plexiform cells are all displaced in one side rather than resting directly on top of the cones, which allow light to pass unimpeded into the cones. These cones have a special structure that aids their detection in detail in the visual image. That is, foveal cones have especially long and slender bodies in contradiction to the much fatter cones located more peripherally in the retina. Also in the foveal region, the blood vessels, ganglion cells, inner nuclear layer of cells and the plexiform layers are displaced to one side rather than resting directly on top of cones, which allows light to pass unimpeded into the cones. Then rods and cones in detail. Diagrammatic representation of rational components of the photoreceptor. Just search for that. The rods are narrower and longer than the cones. But it is not always the case. In the peripheral portion of the retina, the rods are 2 to 5 mm in diameter, whereas the cones are 5 to 8 mm in diameter. In the central part of the retina, in the fovea, there are rods and cones are slender and have a diameter of only 1.5 mm. The major functional segments of either a rod or a cone are shown. So, outer segment, inner segment, the nucleus and the synaptic body. Outer segment, inner segment, nucleus and synaptic body. Outer center, inner center, and outer segment, inner segment, and nucleus, then synaptic body. The light sensitive photochemical is found in the outer segment. In the case of roots, this photochemical is rhodopsin. In the cones, it is one of the three color photochemicals, usually called simply color pigments. Color pigments, which function almost exactly the same as rhodopsin except for difference in spectral sensitivity. Note the large number of discs in the outer segments of roots and cones. Each disc is actually an enfolded shelf of cell membrane. There is as many as 1,000 discs in each rod or cone. Both rhodopsin and the color pigments are conjugated proteins. They are incorporated into the membranes of the disc in the form of transmembrane proteins. The concentration of these photosensitive pigments in the disc are so great that the pigment themselves constitute about 40% of the entire mass of the outer segment. The inner segment of the rod or cone contains usual cytoplasm with cytoplasmic organelles. Especially important are the mitochondria, which are explained later, play an important role in providing energy for function of the photoreceptors. So when we try to learn about this structure. Pigmented layer is the rod, cone region, distal and proximal bipolar cell, vertical pathway, amacrine cell, bipolar cell, horizontal cell, lateral pathway, amacrine cell, ganglionic cell, two optic nerve, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, and ganglion cell layers. Pigmented layer, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, stratum opticum, inner limiting membrane, etc. Once again, pigmented layer, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, stratum opticum, then inner limiting membrane. Pigment epithelium, outer limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, and now fiber layer and inner limiting membrane. Photoreceptors are also there, and horizontal cells, bipolar cells, and amacrine cells are also present. So, schematic drawing of the functional part of the roots and cones. Membrane shells lined with rhodopsin of color pigment, outer segment, mitochondria is also present, then inner segment, outer limiting membrane, and nucleus along with synaptic body. These are the major portions. Then pigment layer of the retina. The black pigment melanin in the pigment layer prevents light reflection throughout the globe of the eyeball, that is extremely important for color vision. Prevents color reflection throughout the globe of the eyeball. Prevents light reflection. This pigment performs the same function in the eye as the black coloring inside the bellows of camera. Without it, light rays would be reflected in all directions within the eyeball, and it would cause diffuse lighting of the retina rather than the normal contrast between the dark and the light spots required for formation of precise images. The importance of melanin in the pigment layer is well illustrated by the absence of this melanin in case of albinos, people who have hereditary lacking in melanin pigment in all parts of their bodies. When the albino enters a bright room, light that impinges on the retina is reflected in all directions inside the eyeball by the unpigmented surface of the retina and by the underlying sclera, a single discrete spot of light that would normally excite only a few rods or cones are reflected everywhere and excite many receptors. Therefore, the visual activity of albinos, even with the best optical corrections, is seldom better than 20 by 100 or 20 by 200 rather than a normal 20/20 values. The pigment layer also stores large quantities of vitamin A. This vitamin A is exchanged back and forth through the cell membranes of the outer pigment of the rods and cones, which themselves are embedded in the pigment. Vitamin E is an important precursor of the photosensitive chemicals of the rods and cones. Then blood supply of the retina, the central retinal artery and the choroid. The nutrient blood supply for the internal layer of retina is derived from the central retinal artery, which divides to supply the entire inside retinal surface. Thus, the inner layer of the retina have their own blood supply independent of the outer structures of the eye. However, the outermost layer of the retina is adherent to the choroid, which is also a highly vascular tissue lying between the retina and the sclera. The outer layers of the retina, especially the outer segments of the rods and cones, depend mainly on the diffusion of the choroid blood vessels for their nutrition, especially for their oxygen. Then retinal detachment. The neural retina occasionally detaches from the pigment epithelium. In some instances, the case of such detachment is injury to the eyeball that allows fluid or blood to collect between the neural retina and pigment of epithelium. Detachment is occasionally caused by contracture of the fine collagenous fibers in the vitreous humor, which pull areas of the retina toward the interior of the globe. Partly because of diffusion across the detachment gap and partly because of independent blood supply to the neural retina through the retinal artery, the detached retina can resist degeneration for days and can become functional again if it is surgically replaced in its normal relation in the pigment epithelium. It is not replaced zone, however, retina will be destroyed and will be unable to function even after surgical repair. Photochemistry of vision. Both rods and cones contain chemicals that decompose on exposure to light and in the process excite the nerve fibers leading from the eye. The light-sensitive chemical in the rods is called rhodopsin, and light-sensitive chemicals in the cones called cone pigments or color pigments. Have compositions only slightly different from that of rhodopsin. In this section, we will discuss principally the photochemistry of rhodopsin, but the same principles can be applied to the cone pigments also. Rhodopsin and retinal visual cycle and the excitation of the rods. Rhodopsin and its decomposition by the light energy. The outer segment of the rod. Rhodopsin and its decomposition by light energy. The outer segment of the rod that projects into the pigment layer of the retina has a concentration of about 40% of the light-sensitive pigment called rhodopsin or visual purple. The substance is a combination of the protein scotopsin and the carotenoid pigment retinal. Rhodopsin, scotopsin and the retinal. Furthermore, the retina is a partial type called a Lens retinal. This form of retinal is important because only this form can bind with scotopsin to synthesize rhodopsin. When light energy is absorbed by rhodopsin, the rhodopsin begins to decompose within a very small fraction of a second. And then because of this rapid decomposition, its photoactivation of electrons in the retinal portion of the rhodopsin, which leads to instantaneous change of the cis form of retinal into all trans form that has same chemical structure as a form but has different physical structure. It is straight molecule rather than an angulated molecule. Because the three-dimensional orientation of the reactive sites of all trans retinal no longer fits the orientation of the reactive sites in the protein scotopsin, the trans retinal begins to pull away from the scotopsin. The immediate product of is bathorhodopsin, which is a particularly split combination of the all trans retinal and scotopsin. Bathorhodopsin is extremely unstable and decays in nanoseconds to lumerhodopsin. This product then decays in microseconds to metarhodopsin. Lumerhodopsin, metarhodopsin, earlier bathorhodopsin. millisecond to metaredoxin 2. Metaredoxin 1 and metaredoxin 2, and finally much more slowly in seconds into the completely split products scotopsin in all transretinal. It is the metarodopsin 2, also called activated rhodopsin, that excites electrical changes in the rods and transmit the visual image into the central nervous system in the form of optic nerve action potential, as we discuss later. So, reformation of rhodopsin. The first stage in the reformation of rhodopsin is the reconvert all transretinal into 11 cisretinal. This process requires metabolic energy and is catalyzed by the enzyme retinal isomerase. Once the 11th of retinal is formed, 11th cis retinal is formed, it automatically recombines with the scotopsin to reform rhodopsin, which then remains stable until its decomposition is again triggered by absorption of light energy. Role of vitamin A for formation of rhodopsin. Note the second chemical molecule by which all transretinal can be converted into 11 cis-retinal. The second route is by conversion of the all transretinal first into all transretinal, which is one of the vitamin A. Then all transretinal is converted into 11 cis-retinal under the influence of the enzyme isomerase. Finally, the 11 cis-retinal is converted into 11 cis-retinal, which comprises the scotopsin to form new rhodopsin. Vitamin A is present both in the cytoplasm of the rods and in the pigment layer of the retina. Therefore, vitamin A is normally always available to form new retinal when needed. Conversely, when there is excess retinal in the retina, it is converted back into vitamin A, thus reducing the amount of light-sensitive pigment in the retina. Interconversion between retinal and vitamin E is especially important in long-term adaptation to the retina at different light intensities. The night blindness. Night blindness occurs in persons with severe vitamin A deficiency. Because without vitamin A, the amounts of retinal and rhodopsin that can be formed is severely depressed. This condition is called night blindness because the amount of light availability at night is too little to permit adequate vision in vitamin A deficient persons. For night blindness to occur, a person usually must remain on a vitamin A deficiency diet for months because large quantities of vitamin A are normally stored in liver and can be made available to the eyes. Once night blindness develops, it can sometimes be reversed in less than 1 hour by intravenous injection of vitamin A. Continuation will be dealt with the next video. Thank you for listening.